Good morning, everyone, and thanks very much to the organisers of EMPSEB for giving me the opportunity to present at this meeting. My name is Susan Johnston, and I'm a research group leader based at the Institute of Evolutionary Biology at the University of Edinburgh. So, a central theme of my research has been to integrate genomic information with long-term field data in order to understand how evolution is acting in the wild. So today I decided to give a bit of an overview of the research that we're doing and how we investigate the genetic basis of traits of interest and how we use this genetic information to examine how these traits are evolving over time. So as evolutionary biologists, one of the key questions we're interested in is why we see so much variation in the world around us. And this is from the broad level of the species down to the level of populations or even individuals. So what we're ultimately interested in is what helps some populations or individuals survive better than others and what helps some reproduce better than others. So the variation that we see in individual phenotype will be the result of the genotypes of those individuals, but also the environment that they experience. And as an evolutionary biologist, I'm particularly interested to know about what the effect of genotype is in contributing to this variation. And this is because traditionally, studies investigating evolution in the wild could only really look at the association between phenotype and fitness. So when I talk about fitness, I'm talking about how well individuals survive and reproduce. And indeed, selection in a population will actually happen at the level of a phenotype. However, in evolutionary terms, we're ultimately interested in what's happening at the level of the genome. And in order to do so, it's important to understand the genetic architecture of traits that contributes to the genetic variation that we have in populations, how this is manifest in phenotypic variation, and ultimately how this is associated with fitness. So what do I mean by the genetic architecture of traits? Well, first of all, it's important to know if a trait is actually heritable, and that is what proportion of the variation that we see is actually attributed to genetic effects. So a high heritability trait suggests that there's a high propensity for the trait to respond to selection, whereas if the trait has a fairly low heritability, so there's very little genetic variation underpinning it, then we expect to see much slower responses to selection, if any. But it's not just thinking about this broad term of how much genetic variation there is, we also want to understand the architecture of that variation. So how many genes underpin the trait of interest, how small or large are their effects on phenotype, do the genes interact with each other or with the environment, and also what is the origin of the genetic variation? Can it arise through things like mutation and migration and so on? So I believe that by determining the genetic architecture of traits and linking this to fitness is really the ultimate goal of helping us to understand evolution in natural populations. However, genomic studies in natural populations have actually been very limited, and this was due to a lack of suitable genomic technologies and model systems. So when I started my PhD in 2006, the only markers that were really available were things like microsatellite markers or sort of low throughput sequencing such as Sanger sequencing. And indeed, for the first 2 years of my PhD were spent developing 12 microsatellite markers for use in a single species. But by the time I finished my PhD in 2010, there'd been an explosion in affordable genomic technologies. And really over the last decade, we've seen that genomic resources like this are tractable in almost all species. So things like really high throughput sequencing to long read sequencing through things like nanopore. But also there have been these concerted efforts to try and get genomic information for as many species as possible in creating these huge public resources. So indeed, at the end of my PhD, I was using these SNP arrays to type tens of thousands of single nucleotide polymorphisms throughout the genome. So now that we have these genomic technologies, but we also have information from long-term studies and through like long-term ecological studies in the wild, there's 4 questions that my research really aims to answer. So first of all, are traits heritable? So what proportion of the variation that we see is due to genetic effects? Secondly, if so, what's the genetic architecture of this heritable variation? Thirdly, is this genetic architecture associated with fitness? And finally, is this genetic architecture actually changing and therefore evolving over time? So in this talk, I'm going to focus on two study systems that I've been working on for a while. And both of these systems are overseen by Professor Josephine Pemberton at the University of Edinburgh, who's been a long-term collaborator of mine. So the first population that I'll talk about is the soy sheep of St Kilda. So these are a Neolithic breed of domestic sheep that are endemic to the St Kilda archipelago out here in the northwest coast of Scotland. So this is about 160 kilometres west of the mainland. So these sheep are thought to have arrived on St Kilda around 4,000 years ago. And it's the individuals that live in the Village Bay area of the population here that have been subject to an intensive study since 1985. And indeed, we have data from more than 10,000 sheep collected over this period. And the second population that I'll talk about is red deer on the island of Rum. So this is an island in the Inner Hebrides of Scotland, still quite isolated. And it's the individuals that live in the northern part of this island that have been studied on an individual basis since 1971. And we have data for more than 5,000 deer over this period. So these populations are quite tractable for investigating the genetic architecture of traits, and they have sort of three components to help us understand trait evolution. The first is that we have extensive life history and phenotype information from birth until death for most of these individuals. So we go out on these expeditions throughout the year, and this is an example of my colleague Camilo Brenos weighing a soy sheep in the field. Secondly, we have really accurate and comprehensive pedigree information, and this is really important for our statistical models, but also for determining individual reproductive success. And we've actually developed our packages, for example, by my colleague Jiska Hausman, that are building accurate and comprehensive pedigrees using single nucleotide polymorphism information. And finally, most of the individuals in these studies have been genotyped on these Illumina SNP chips that are species specific. And at the moment, we have 39,000 SNPs genotyped in 8,000 sheep and 38,000 SNPs typed in 3,000 deer. So the first case study that I'd like to share with you is really probably the most complete analysis that we've done in any of these populations. And this is what my PhD was looking at, which is the horn morphology in the soy sheep of St Kilda. So this is an example of a male soy sheep, and you can see that they have these nice big horns that are really important in use for competing for access to mates. So soy sheep actually have one of the most promiscuous mating systems of any mammal, and they have really strong intra-male competition for access to estrus females during the rut. So what happens is they form a loose dominance hierarchy, but then when they find a female who's an estrus, generally males that are evenly matched will do things like this parallel walking, and then they'll usually come to blows with each other and fight each other. And generally it tends to be the males that win these fights, or males that are at the top of this dominance hierarchy, that will hold the females in consort while they're an estrus, whereas males that generally have smaller horns can only really watch and hope for the opportunity for some sort of sneaky mating to happen. So perhaps unsurprisingly, males with larger horns in the population tend to sire more offspring. However, this actually leads to a bit of an evolutionary puzzle within the population. So of course we have these really dominant males that have these really large and they have these really long and sort of thick horns. But then we see a number of males within the population that have fairly modest horns or actually develop fairly small horns. So I would say that these males are actually of a similar age. And then we have, even more interestingly, some males that don't develop any horns at all. So these two males here have what's known as scurs. And what's happened is that the bony core of the horn hasn't developed at all, we only have the outer keratin sheath. So these are really sort of fragile stumps on the head. And occasionally we even have males that don't develop any horns whatsoever. So the big question is that if bigger horns are better, then why do these small horns persist within the population? So the whole sort of premise of the work that's done in my group really hinges on a single statistical framework. And this is the animal model framework that was originally developed for animal breeding. So this is the sort of equation of the model that we use, and I'm going to talk you all through it. But this is actually sort of conceptually a model that many of you will already be using in your research. So it's a generalized linear mixed model. So we'll have our response variable here. So for example, we can have horn length, and then we'll fit fixed effects in the model that affect the mean of the trait. So in this case, we fit things like the capture age when we measure the horn and also the capture season. We also look at things like we can fit another random effect, which is the additive genetic effect, and this calculates the heritability. And we do this by fitting a genomic relatedness matrix that's built between all the individuals using their genomic data. And we can use this to see if individuals that are more sort of genetically similar are more phenotypically similar, but this will also sort of mop up the variation due to, relatedness within the population. And it's essentially almost looking at a correlation between the two, and the strength of the correlation between phenotype and relatedness is conceptually very similar to what the heritability will be. We then fit these additional random effects, so things like individual identity to account for pseudo-replication, the birth year and the capture year, which are common environment effects, which may also affect the phenotype. And then also we have this error term here. So this actually will give us the broader idea of the genetic architecture through determining the heritability of the trait. But then there's a key adaptation that we can use to try and understand the underlying genetic architecture. And this is by looking at the effect of individual SNP genotypes throughout the genome and seeing what the significance of these genotypes are and the effect sizes of these as an additional fixed effect. So essentially what we do is we take this model, and if we have 39,000 SNPs, we essentially run this model 39,000 times with SNP genotypes at different genomic regions fit as fixed effects. And this is essentially what's known as a genome-wide association study. So to show this in practice, so here is the heritability and a genome-wide association study looking at both horn length and horn circumference within this population. So we find that both of these are heritable, so about 30% of the variation in horn length and 41% of the variation in horn circumference is due to genetic effects, so it is heritable. And then here is the results of our genome-wide association study. So each point indicates a different SNP, and on the y-axis this is essentially a measure of significance of each of these SNP models. And anything that goes above this dashed line is significantly associated with horn size. And you can see that we have this big peak on chromosome 10, and this corresponds to the locus RXFP2, and this is really strongly associated with horn size in males. Indeed, it explains almost all of the variation that we see in this phenotype. So we were able to use this information to develop a genetic test for Horn's genotype, so the genotype of RXFP2. And we found this quite sort of interesting segregation pattern. So we find that individuals that have the wild type allele tend to have these very long and thick horns. Individuals that are heterozygous at this locus have more modest horn development. And males that are homozygous for what we call the polled allele either develop very small normal horns or they don't develop any horns at all. So at this point, we still don't know what distinguishes, what drives this difference between these two sort of genetically identical but phenotypically different males. But essentially, we can see that having more copies of the plus allele gives you larger and longer horns, and more copies of the P allele essentially gives you smaller or potentially absence of horns. So now that we knew the genotype, we wanted to see if this was associated with individual fitness. So we looked at the difference between the reproductive success in a given year of males with the three different genotypes. And we found that males that were homozygous for the P allele, so essentially the smaller horned males, have much fewer offspring. However, the plus males and the heterozygous males actually had more or less the same number of offspring as each other in a given year. But when we looked at survival within a given year, we found that males that were homozygous for the plus allele have much reduced survival. But we found that the heterozygous and homozygous P males actually had very similar survival to each other. So when we took a composite measure of fitness, so this integrates the reproductive success and the survival of the individual into one measure, we found that the heterozygous males within the population have the highest fitness. And essentially, this variation in horn type is maintained by a heterozygote advantage at the horn's locus. So one measure of evolution within a population is trying to determine if allele frequencies are actually changing over time. So we did find that in this population, the large horned allele is actually decreasing in frequency. So over a 22-year period, or possibly more than that, we found that there was a decrease from about 0.54 to below 0.5. And we wanted to determine if this change in allele frequency actually differs from what we would expect due to chance or due to genetic drift. So this is because within a population such as the soy sheep, it's not a sort of like Fisher-Wright, Hardy-Weinberg equilibrium kind of population. We actually have a lot of variance in mating success, and we also have periodic population crashes, and this can actually influence allele frequencies and how they change over time. So in order to bypass this, we used a gene drop approach using an R package that was developed by Darren Hunter. And what we do is we sample alleles in the founder cohorts, and then we allow them to sort of transmit through the pedigree, sampling them 50-50 from the parents. And this allows us to get a null distribution of how allele frequencies will change purely under drift. And then we can compare what we see in the real world and what we observe through the simulations to see if there's any sort of directional selection on this locus. So this here is the line that we find with the sort of our observed line. So this is in here in red. And then all of these black lines that are appearing are basically each of the individual gene drop simulations. And we can look at the slope of this line and compare it with the slope of the other lines. And this is what we see here. And the slope of our observed line actually fits within the sort of range that we would expect under drift. So this is actually kind of consistent with the fact that there's heterozygote advantage at this locus. So we might expect it to be under balancing selection rather than under directional selection. And we do see that when we look at these kind of changes in allele frequency, that they are slightly less than what we would expect due to chance. But really the large horned allele frequency is stable over a 30-year period in this population. And actually the equilibrium frequency that we see is actually around 0.5. So the thing is linking genotype to fitness in this sense is really useful for trying to understand evolution at the locus, but it still kind of neglects to look at what's actually happening in the real world and how is this variation in life history manifested. So an undergraduate honours student, Sarah Dobson, last year was actually looking at differences in behaviour between the 3 different genotypes. And what her research suggests is that males that have these larger horns, or they're homozygous for the plus allele, they're actually fighting more and at a much younger age. And this is actually leading to higher mortality. Whereas males that are sort of homozygous for the P allele are actually continuing to feed during the rut, which the homozygous plus males don't. And they're also searching for unguarded females to mate with, so they do achieve some matings. And if you're heterozygous, you're actually kind of getting a bit of the best of both worlds. So you're competitive enough to fight, and you're more competitive than these kind of hornless males, but also you will take some time to feed, and you will get sort of more of these sort of sneaky opportunities as well. So really the variation in life history is manifest by this trade-off within the population that benefits the heterozygous males the best. So I'm now going to move on to a related phenotype, but where we see quite a different pattern. And this is in antler morphology in rum red deer. So deer have these antlers that are again really important for intramale competition, and males that are sort of better in this competition tend to hold harems of females. However, work by my colleague Luska Crook, who spent a lot of time looking at antlers within this population, she published a very influential paper in the early 2000s that showed that there's phenotypic selection on antler weight within this population and on the number of points, but actually these traits are not evolving over time despite this selection. So there is genetic variation in antler morphology, but it's being completely maintained within the population. So in order to try and understand this in a bit more detail, a PhD student in my group, Lucy Peters, has been investigating the antler morphology in the rum red deer and its genetic architecture. So instead of just taking one or two phenotypes, we actually used information from a lot of different aspects of the antler. So from the antler weight, but also things like the different dimensions and the number of points. And we wanted to see if the genetic architecture of antlers can shed a bit more light on the phenomenon that we observe. So what Lucy kind of found is that all of the antler measures are actually heritable. So there is substantial genetic variation, and also they're all positively correlated with each other. That doesn't imply that there could be many sort of evolutionary constraints within the antler morphology itself. But when she went to do genome-wide association studies, she did it on all 10 of these traits. And if you remember from the graph before, this is a sort of threshold of significance, and this is the significance here. But Lucy essentially found that there was no regions of the genome that had a fairly large effect on the phenotype. And it does suggest that the genetic architecture of this trait, or these traits, is polygenic, so it's driven by many loci of small effect. However, when looking in small populations, we do sometimes have an issue of power. So it may be that Lucy didn't have the power in the data to actually detect these loci of large effect. So actually she used an alternative approach to genome-wide association study called an empirical Bayes approach. And what this does is it takes the information that we got from the genome-wide association study, so the effect sizes at each of these SNP and also the p-values, and it can estimate the distribution of effect sizes underlying the phenotype, and also what we call the false discovery rate. So how many of these are actually, likely to be associated with phenotype, even though we don't see significant ones looking at the SNPs individually. And what she found is that about 900 SNPs, so about 2% of the SNPs on our SNP chip, had non-zero effects on phenotype, so they are likely to drive some of the variation in antler morphology. So the fact that the trait is polygenic, it does make it very difficult to pin it down to a specific driver that explains this lack of evolution, but we can sort of offer some more likely explanations. So it may be that having so many loci underpinning this antler morphology means that there's a very large mutational target. So there could be more opportunities for new mutations to arise in these loci and therefore kind of maintain the genetic variation that we see. It also may be that because selection is distributed across so many loci, that the selection at a given locus is weak, and this actually cannot overcome the effects of drift within this population. So really it's only when selection is very strong at individual loci that we can actually see this change. And also it could be that there's some sort of pleiotropic effects. So some of these loci may affect other traits under selection, or they're in linkage disequilibrium with traits that are under different types of selection. And it could be that there's lots of trade-offs occurring in fitness across the genome. So this is actually still the subject of ongoing work by Lucy, but what we can do is look at things like the genomic correlation with fitness and so on. And this is the kind of work that she's also doing just now. So through the work of myself and also Camilo Brenos, who you saw earlier doing some sheep weighing, who was also a postdoc at the same time as me on this project, and also through the work of PhD students such as Alex Sparks and Lucy Peters, we've been looking at the genetic architecture of different traits, but also finding that this can either be due to few large effect loci and also small effect loci, and somewhere on a spectrum in between. So we have things like the horn size that are essentially driven by variation at a single locus. But then we have other traits that are heritable, such as antibody levels, leg length, and things like recombination rate, where there are some genes of large effect, but also a polygenic component, so we can't determine the full genetic architecture. But then there's other traits, such as vitamin D, body weight, telomere length, and the antler morphology, that appear to be driven by many loci of small effect throughout the genome. Now, with the exception of horn size, despite the fact that we, imagine that these traits could be under selection, we actually don't really see any association of genetic variance and fitness at these other traits. So in some ways it's a little bit unsatisfying, but we've been thinking about other approaches that we can use to try and understand evolution in these populations. So you'll remember at the beginning of this talk, I was talking about this sort of cascade from the genetic variation through to the phenotype and fitness. But it may be that by concentrating on phenotype alone, we're actually sort of missing really the sort of bigger nuances and bigger phenomenon that could be happening in a population, because we're not looking at phenotypes that have, the phenotypes that are under selection or driving variation are things that we actually haven't measured. So I think that we have to think about ways that we can escape the tyranny of the phenotype. And one alternative to doing this is actually just to skip the phenotype and try to determine, rather than the loci underpinning a phenotype that's under selection, just determining which loci are actually under selection. So there's different approaches that we'll use. So I did talk about the allele frequencies changing over time, but for the interests of time here, I'm not going to discuss this anymore. So this was through this gene drop analysis. But another thing that we are doing in both of these systems is trying to determine if there are loci that show signatures of selection over the life cycle of the individual. So we can do this by using a selection component analysis. So this work is actually heavily inspired by another colleague, Nancy Chen, at the University of Rochester, and she published a really nice paper on this in Florida scrub jays in 2019, and highly recommend that you check that out. But one thing that's kind of pivotal to her research and really inspired our research is looking at how selection can act at different stages of the life cycle. So for example, if you think about the gamete, there's essentially selection on the gamete to really make it to the zygote through things like haploid selection. If we think about when you get the zygote, are these individuals actually going to survive through till adulthood? So this is essentially viability selection occurring. When they're adults, do they actually have the ability to become parents and sire offspring? So we can think of this in terms of sexual selection. And then when they're actually parents, trying to think about what kind of the quality of the gametes that they're producing that are actually going on to produce successful zygotes, and thinking about fecundity selection. So in our group, we're really focusing on this gametic selection and viability selection at the moment, to try and understand what kind of selection can be occurring at alleles and the gametes that are transmitted from mothers and from fathers, so from parents, into the actual birth of their offspring. And because we're quite interested in sexual conflict in my lab, we're really focusing at this from the perspective of sex, both from the parental side and from sex differences in offspring mortality, so the inheritance of alleles from different sexes. So we're wanting to investigate if parents of different sexes are more likely to transmit a particular allele, so this indicates some selection at the gametic stage, or if offspring of different sexes inherit one allele more often, so selection at the viability stage. And this work has been done by Anna and Lucy, and in collaboration with Darren Hunter. So the approach that we're using is looking at transmission distortion. So in a nutshell, this is really trying to look to see if there's differences in what alleles are transmitted from a parent. And at a simple level, although our models are a little bit more complicated than this, we can think that if we have, for example, a father who's heterozygous, who mates with a mother who's homozygous at a given locus, the offspring are either AA or AB, so we can see which allele was inherited from the father. And if we look at this across populations, we can look, for example, from the perspective of a father, if they're more likely to transmit an A or B allele. So you can see across 400 gametes that we can characterise, there's not really any difference in which allele is transmitted. But from the female perspective, we can see that, you know, out of 400 matings or so, that around 256 offspring, she'll transmit, the B allele 256 for every 142 times she transmits the A allele. So this can indicate some sort of form of gametic selection. So I'll just quickly present results from some work done by Anna. And she found that when looking at the inheritance of particular alleles into female or male offspring, that there's some evidence of selection in male soy sheep lambs. So she found 4 genomic regions that are associated with distortion in male offspring. So there's one allele that tends to end up in surviving male offspring more than others. And one of these actually corresponds to a prolificacy gene, which means the number of offspring that a sheep has in domestic sheep, so this KLF5. And we're now sort of refining our approaches and integrating it with gene drop information and developing null models to try and refine this to look at more life history stages in both the sheep and the deer. So just to finish up, I think that through this talk, I hope that I've shown you that determining the genetic architecture of traits can really help give us clues into their contemporary evolution. But also, without phenotype, long-term data can actually be leveraged to look at signatures of selection over time, and particularly these differences in allelic transmission between generations. And I think that different aspects of what we've done are really adaptable to different systems. And indeed, if any of you feel inspired by any of the work that I've presented today, please don't hesitate to get in touch. I can chat now during this meeting or really at any other time that's convenient for you. So with that, I just want to acknowledge that I'm a small cog in a big machine, and this work's been overseen by Josephine, John and Jill, either through the genomic work and also in the field work. And also I've had the honour of collaborating with many different people to do these studies, and with lots of funders and different institutes. And I'd really like to thank you all for your attention.